So I'm a postdoc in the ARIS group at the Australian National University, and I'll be presenting to you a software called RISER to enrich or deplete classes of RNA during direct RNA sequencing. Inside cells, there are many different classes of RNA which vary in their structure, function, and role in disease. They also vary in their abundance, and it's still a major challenge to characterise RNAs of low abundance, particularly in the presence of highly abundant RNA. So as an example, long non-coding RNAs typically have a low but highly tissue-specific expression pattern, and this can be obscured in RNA measurements by messenger RNA and other highly abundant classes. Biochemical approaches can be used to enrich or deplete specific RNA molecules before sequencing, but the problem with these is that they're time-consuming, expensive, they risk degrading the RNA and also introducing bias into your sequencing results. Nanopore sequencing allows the direct measurement of RNA. The way that it works is individual strands get pulled through a pore while an electrical current signal gets recorded. This can then be decoded to determine features of the RNA, such as its nucleotide sequence or the presence of biochemical modifications. However, the nanopore direct RNA sequencing platforms have limited throughput and the pores degrade over time. And the problem with this is that that limits the sequencing capacity. So it's really important that this limited sequencing capacity is focused on the RNA of interest throughout the sequencing run. A unique advantage of nanopore sequencing is the adaptive sampling feature. So this is where in real time signals get base called and mapped against a list of targets. And if the strand does not match the target, it simply gets flicked back out from the pore by reversing the pore voltage. The problem, however, with this approach is that the targets must be specific sequences that are known beforehand. And this is a problem when you're dealing with samples or organisms with incomplete references. The other issue is base calling and mapping are expensive computations. And this is a problem because it's really important to be making decisions as quickly as possible for adaptive sampling to be beneficial. To overcome these limitations, we developed RISER. RISER uses a neural network to directly classify the start of the RNA signal and then sends an accept or reject command to the sequencing hardware depending on the user-defined target class. This focuses the limited operating time of the nanopores on the RNA class of interest. The premise for how it works is since RNA is always sequenced in the 3' to 5' direction of the molecule, the 3' end of the RNA is always encoded at the start of the nanopore signal. So the RISER classification relies on being able to distinguish between different RNA classes from their 3' ends. So the first target class that we chose was messenger RNA, which is highly abundant in the majority of samples. mRNAs also have distinct features in their 3' UTR that are not in non-coding RNAs. So we expect these to be distinguishable from the start of the nanopore signals. So to design RISER, we prioritise both accuracy and real-time efficiency. We found that using 4 seconds as a maximum input length would allow RISER to impact as many molecules as possible while also retaining enough information to make a good prediction. We next systematically optimise the hyperparameters of 3 convolutional architectures, which are known to perform well for handling the variance and also the local signal patterns in nanopore signals. So on the held out test dataset, the ResNet and CNN outperform the TCN at all input lengths with respect to accuracy, precision, and true and false positive rates. But since this is a real-time application, it's really important that we're making decisions as quickly as possible. So we selected the CNN as our architecture since it performs around twice as fast as the other models. So this is the architecture of the RISER model. The input are the raw nanopore signals and the output are the probabilities that the molecule belongs to the target class or not. If the predicted probability is confident enough, then RISA will send an accept or reject decision to the sequencing hardware. Otherwise, it will retry once more of the molecule has passed through the pore. We tested the generalizability of the model using an independent data set from a cell line that hadn't been used for any training or hyperparameter tuning. The model achieved a really strong accuracy of 94%, as well as a high true positive and low false positive rate. Per RNA biotype, the accuracy was also over 95% for all biotypes except link RNAs. We looked into what was happening here, and we found that over 80% of the mispredicted link RNAs actually shared 3' ends with mRNA, which is why it's not possible to discriminate them. This performance was also recapitulated on a separate independent test set from another cell line, and also that was sequenced by a different lab. So RISA can be easily retrained for other targets. So we did this for mitochondrial RNA since it's highly abundant in cells with high energy demands, such as muscle. We repeated the same set of independent tests on the mitochondrial RNA model and found that it performed even better than the messenger RNA model, and also achieved strong accuracy across the various biotypes. So having verified the strong performance of the models, we then ran RISA in real time during live MinION sequencing of poly-A-selected RNA from HEC293 cells. To avoid flow cell bias, we tested both the RISA and control conditions in the single flow cell. The way that we did this was we split the flow cell channels into 2 groups in the code. And in one group, we set RISA to deplete both messenger and mitochondrial RNA, and we used no RISA in the other half as a control. We also repeated this same setup with a separate flow cell, but with both halves of the flow cell being used as a control. And the purpose of this was to allow us to assess the impact of RISA on read counts versus the natural variability in read counts that occurs during nanopore sequencing. So as you can see in orange, RISA reduced the read counts of both messenger and mitochondrial RNA by around 90% on average. And what we found interesting was that as a result of this depletion, it allowed link RNAs to be increased by around 50% on average. This was considering those that don't share 3' ends with mRNA. These effects were statistically significant compared to the purple control group. And importantly, RISA mistakenly accepted only 1% of mitochondrial RNA reads and 5% of mRNA reads. These results were also recapitulated when we considered a second control experiment. So we also tested the hypothesis that RISA would be more efficient than sequence-based adaptive sampling because it uses a simple binary classification straight on the raw signal rather than base calling and mapping. So using a test set of 1,000 fixed-length signals, we found that RISA with a GPU was over 40 times faster than sequence-based adaptive sampling, and even faster than that when using just a CPU. RISA also only needed 4 seconds worth of signal to correctly identify the RNA class, while adaptive sampling required up to 46 seconds to identify it. So to summarize, RISA is much faster, can be run with just a CPU, and also requires less input signal to identify the targets compared to sequence-based adaptive sampling with base calling and mapping. So the last thing I wanted to show you was a clinically interesting application of RISA. Globin messenger RNA dominates whole blood samples, which makes it difficult to obtain a good coverage for other clinically interesting transcripts. So existing globin depletion methods are biochemical based, and the problem with this is that fragments the RNA, and so there's no globin depletion method that's compatible with long-read direct RNA sequencing. So what we did was retrained RISA to detect globin messenger RNA. And you can see here on a held out test data set, the performance was very strong. So we then took it into a live sequencing run on a whole blood sample and found that RISA reduced the read count for globin transcripts by over 90% on average, which resulted in non-globin messenger RNA increasing by around 16% on average. So the reason it was so easy to update RISA for these other classes was that it was designed to be modular and extensible. So it's easy to replace the model or adapt the signal processing approach, or if Nanopore update their read until API, that's just encapsulated in a wrapper class. So the changes are isolated. So we've developed a simple command line tool to run RISA. You only need to specify your target class, whether you want to enrich or deplete it, how long you want to run RISA for, and what sequencing kit you're using. So we support both the RNA 002 and the new RNA 004 sequencing kits. You don't need to change anything when setting up your run. RISA will just interact with the sequencer in the background. So to summarize, RISA is the first biochemical-free technology for real-time enrichment or depletion of classes of RNA during direct RNA sequencing. It alleviates the limitations of biochemical enrichment and provides a new type of sequencing control. It's available now on GitHub, and there's a paper if anyone wants to see any more details. So with that, I'd like to thank the rest of the RISA team and in particular my supervisor, Eduardo. Happy to take any questions.
Great talk. Quick question for you. So a lot of link RNAs, they are the antisense transcripts, right? So how the RISA can distinguish from the antisense link RNA and the protein coding counterparts?
Sorry, it's really hard to understand with the
right,
microphone. Maybe just out, yeah.
Use the microphone. Yeah. So many link RNAs, they are antisense transcripts. Right? So how can the RISA distinguish between antisense transcript and the layer of protein coding counterparts?
So RISA is not distinguishing the link RNAs. It's distinguishing whether or not it's messenger RNA. So if the link RNA shares a 3' end with the mRNA, then it won't be possible to distinguish them, to say that it's not mRNA. But if the link RNA is from a non-coding region, it will, then RISA will say it's not an mRNA. Yeah, cool, no worries.
Okay, do you have other questions? Okay, it's quite surprising. Only 4 seconds they can tell between whether they are mRNA or not. So yes. For those 4 seconds, how many base pairs are already used in your analysis?
So for the RNA 002 kit, which is the old one, that translocates at roughly 70 bases on average. So it's around 280 nucleotides for 4 seconds. But the new kit is much faster. It's 130 bases per second. And we don't actually take 4 seconds for the RNA 004 kit. We take much less because we only need like 280 nucleotides worth. But we're not working in sequence space. We're working directly with the signals.
Oh, okay. So 200 is enough.